Good morning friends, my topic is phylogenetic studies, phenetic classification and Bergey's manual. Phylogenetic studies. Microbial phylogenetics is a study of the manner in which various groups of microorganisms are genetically related. So, microbial phylogenetics is a study of the manner in which various microorganisms are genetically related. This helps to trace their evolution. To determine the phylogeny of an organism, scientists must determine whether a similarity is homologous or analogous. So, this will help to trace their evolution and to determine the phylogeny of an organism. Scientists will determine whether that similarity is homologous or analogous. The advancement of DNA technology, the area of molecular systematics, describes the use of information on the molecular level, including DNA analysis. Categories within taxonomy classification are arranged in increasing specificity. The most general category in taxonomy classification is domain, which is a point of origin of all species. All species belonging to one of these domains, that is bacteria, archaea and eukarya. So the categories within taxonomy classification will be arranged in increasing specificity, and most general category in this classification is a domain. It is a point of origin of all species, and all species will belong to one of these domains, that is either bacteria, archaea or eukarya. In each of the three domains, we find kingdoms, the second category within taxonomy classification, followed by subsequent categories that include phylum, class, order, family, genus, and species. We already discussed that taxonomic classification are arranged in increasing specificity. Then first we discussed about the origin, that is domain, and within these three domains, we will find kingdoms, the second category within taxonomy classification. Then it will be followed by subsequent categories such as phylum, class, order, family, genus, and species. At each classification category, organisms become more similar because they are more closely related. So, at each successive classification category, organism will become more and more similar because they will be more closely related. Phylogenetic classification is concerned with grouping individual species into evolutionary categories. So, phylogeny means we already know it is evolutionary classification. Since the early 1980s, phylogenetic classification has been made much more facile by the invention of molecular taxonomy. The evolutionary classification of organism was based on the nucleotide sequence divergence at individual loci. So, the evolutionary system of classification, that is phylogenetic classification, is based on the nucleotide sequence divergence at individual loci, that is genes. The term phylogeny means Greek phylon, that is tribe or race. Genesis means generation or origin. It refers to the evolutionary development of a species. After publication of Darwin's On the Origin of Species, that is in 1859, biologists began trying to develop phylogenetic or phyletic classification systems. The system is based on the evolutionary relationship rather than general resemblance. So, the system, that is phylogenetic system, is based on the evolutionary relationship rather than genetic resemblance. This has proven difficult for bacteria and other microorganisms, primarily because of the lack of a good fossil record. So, it was difficult to trace its ancestors, and it became difficult for bacteria and other microorganisms. This The direct comparisons of genetic material and gene products such as RNA and protein overcome some of these problems. So, these problems can be overcome by direct comparisons of genetic material such as RNA and proteins. A phylogenetic tree is a diagram used to reflect evolutionary relationships among organism or group of organism. Notice in the rooted phylogenetic tree that three domains, bacteria, archaea and eukarya, diverge from a single point and branch off. So, a phylogenetic tree is a diagram used to reflect the evolutionary relationship among organism or microorganism. In a phylogenetic tree, there will be three domains, bacteria, archaea and eukarya, and it will be diverging from a single point, and it will be branching off. Phenetic classification. Phonetics is an attempt to classify organism based on overall similarity, usually by morphology or other observable traits, regardless of their phylogeny or evolutionary relationship. It includes various forms of clustering and ordination. Phenetic classification of organism is based on measurable similarities and differences rather than the genetic makeup and evolutionary descendants. So, phenetic classification is an attempt to classify organism based on overall similarity. Usually, it is done based on morphology rather than phylogeny or evolutionary relationship. Phenetic system. Phenetic system is a group of organisms which groups together based on the mutual similarity of the phenotypic characters. So, phenetic system is a grouping of organisms together based on the mutual similarity of their phenotypic characters, that is by comparing their morphologic characters. This classification system succeed in bringing order to biological diversity and clarify the function of morphological structures. For example, motility and flagella are always associated in particular microorganism. It is reasonable to suppose that flagella are evolved in at least some type of motility. That is an example for phenetic classification. Although phenetic studies can reveal possible evolutionary relationship, they are not dependent on phylogenetic analysis. So it is not based on evolutionary relationships. The best phenetic classification is one constructed by comparing as many attributes as possible. Organisms sharing many characters make up a single group or taxon. So it will be done by comparing as many attributes as possible, and this organism sharing many characters will make up a single group or taxon. Phenogram, a diagram depicting taxonomic relationships among organisms based on overall similarity of many characteristics without regard to evolutionary history or assumed significance of specific characters, usually generated by computer. So, phenogram is a diagram generated by computer and it is depicting taxonomic relations among organisms based on overall similarity of the characters, that is it will not depend on evolutionary relationship. Those organisms most similar are classified more closely together. So more closely related organism will be grouped together. Microscopic morphology, cell morphology, rod, coccus or spirillum. So, microscopic morphology include cell morphology, that is rod, coccus or spirillum. Cell arrangement, that is whether it is diplococcus, streptococcus, tetrad, irregular clusters, etc. And special cell structures include flagellum, cilia, spore, capsule. We discussed earlier about flagellum and that is associated with motility. Likewise, this is a microscopic morphology associated with phenetic characters. Phenotypic characters of taxonomic value. Major category, morphology. Its components include colony morphology, Gram reaction, cell size and shape, pattern of flagellum, presence of spores, inclusion bodies, stalks or appendages. It is all included in morphology. That is colony morphology such as Gram reaction, cell size, pattern of flagellum, presence of or absence of spores, stalks or appendages, etc. Motility, non-motile, gliding motility, swimming, swarming, motile by gas vesicles. Such as, likewise, motility can also be used as a phenotypic character. Metabolism, mechanism of energy conservation, utilization of individual carbon, nitrogen or sulfur compounds, fermentation of sugars, nitrogen fixation, growth factor requirements. Such components are included in metabolism. Bergey's Manual. Bergey's Manual of Systematic Bacteriology is a compendium of standard and molecular information about the available prokaryote. It is a compendium of standard and molecular information about the all available prokaryotes. The characteristics used to define sections are normally features such as general shape and morphology, gram staining properties, oxygen relationship, mortality, the presence of endospore, the mode of energy production and so on. The characters used to define this sections will be based on the shape and morphology, gram staining property, oxygen relationship, mortality, presence of endospores, mode of energy production, etc. Bacterial groups are divided into 4 volumes in the following manner. Is gram negative bacteria of general, medical or industrial importance. B is gram positive bacteria with a distinctive property, cyanobacteria and archibacteria. C include actinomycetes, that is gram-positive filamentous bacteria. Bergey's manual is the most widely accepted document used for classification and identification of bacteria. Bergey's manual is the most widely accepted document used for the classification and identification of bacteria. Though the new edition of Bergey's manual is organized along phenetic and utilitarian lines, R.G.E. Murray, in a theoretical introductory article to the manual, suggests that the kingdom prokaryote can be divided into 4 divisions and 7 classes. The 4 divisions are characterized primarily as follows: the presence or absence of cell walls, the chemical and structural nature of the cell wall. The first division of Bergey's manual of systematic bacteriology consists of 4 volumes. So R.G.E. Murray, in a theoretical introductory article to the manual, suggests that kingdom prokaryote can be divided into 4 divisions and 7 classes. The 4 divisions are characterized primarily as follows: the presence or absence of cell walls, the chemical and structural nature of the cell wall. The first edition of Bergey's manual of systemic bacteriology consists of 4 volumes. Volume 1 include gram-positive bacteria, Volume 2 gram-negative bacteria, Volume 3 bacteria with unusual properties including archaea, Volume 4 filamentous bacteria, and Volume 5 plankomycetes, spirochetes, bacteriotes, fusobacteria. The system does not have much phylogenetic information. So there are different volumes for Bergey's manual. Volume 1 include gram-positive bacteria, Volume 2 include gram-negative bacteria, Volume 3 include bacteria with unusual properties including archaea, Volume 4 include filamentous bacteria, 5 include spirochetes, planomycetes, bacteriotes, etc. And this do not have much phylogenetic information. The Bergey's manual are also characterized by presence or absence of cell walls, chemical and structural nature of the cell wall.